Welcome to Animal Biotechnology. Today, we're going to explore one of the most fascinating and powerful techniques in genetic engineering, the retroviral method. Imagine being able to rewrite the genetic code of a living organism by hijacking the very pathogens that usually cause disease. This method does exactly that, turning a natural enemy into a microscopic delivery truck for beneficial genes. Let us dive in and see how this incredible process works. To understand how this works, imagine you need to deliver a highly sensitive package into a heavily guarded fortress. If you just walk up to the gate, you will be turned away. But what if you disguise your package inside a Trojan horse that the guards are already programmed to let in? In animal biotechnology, the fortress is the nucleus of an animal cell, and the Trojan horse is a virus. By scooping out the harmful, disease-causing genes of a retrovirus and replacing them with our beneficial gene of interest, we create a highly efficient, microscopic delivery truck. Now that we understand the basic analogy, let us look at the actual biological machinery at play. A retrovirus is a specific type of RNA virus. When it enters a host cell, it uses an enzyme called reverse transcriptase to convert its RNA genome into DNA. Then, another viral enzyme called integrase permanently stitches this newly synthesized viral DNA into the host cell's own genome. For our purposes, scientists engineer these viruses to be replication defective. This means the virus can successfully infect a cell and insert our desired gene, but it completely lacks the ability to multiply and cause an ongoing infection. Building on that biological mechanism, you might wonder why we prefer this over older techniques. Before retroviral vectors, scientists relied heavily on DNA microinjection, which is incredibly tedious and often results in very low success rates. The retroviral method is a massive upgrade because it offers a much higher efficiency of gene transfer. Furthermore, retroviruses typically insert exactly one copy of the gene into the host DNA. This is crucial because it prevents the chaotic, multi-copy insertions that can disrupt normal cellular functions and cause unwanted mutations. This reliability makes it a foundational technology for modern gene therapy. Let us bring all these concepts together and visualize the entire process. On the screen, you can see the complete journey of our engineered retrovirus. Starting on the left, the harmful genes are removed and replaced with our RNA transgene. This modified virus is then introduced to an 8-cell stage embryo in a petri dish. Notice the magnified view of the cell. The viral RNA undergoes reverse transcription into DNA, which then integrates directly into the host chromosome. Finally, these treated embryos are implanted into a surrogate mother mouse, which eventually gives birth to chimeric offspring, shown on the far right with patchy fur, indicating the new gene is present in some of its tissues. Having seen the physical process, we must now address a critical experimental challenge. To successfully infect embryos without overwhelming and killing them, scientists cannot just guess how much virus to use. They must carefully calculate a metric called the multiplicity of infection, or MOI. This calculation is absolutely essential because it ensures that there are enough viral particles present to guarantee successful gene transfer, but not so many that the embryonic cells suffer from toxicity and die. It is a delicate balancing act between efficiency and cell survival. This delicate balance brings us to the mathematical representation of the multiplicity of infection. The formula is M equals C times V divided by N. Let us break down what each symbol means. M represents the multiplicity of infection, which is the average number of viral particles attacking each individual cell. C stands for the viral titer, or the concentration of infectious particles per milliliter. V is the volume of the viral suspension added, measured in milliliters. Finally, N represents the total number of target embryonic cells. If you increase the concentration or the volume, the total number of viruses goes up, leading to a higher M. Conversely, if you have more cells but the same amount of virus, M decreases. Now that we have our formula, let us apply it to a real laboratory scenario. A researcher wants to infect 10,000 early embryonic cells. They want an MOI of 5, meaning 5 viral particles per cell, and their viral stock has a concentration of 1 million particles per milliliter. We need to find the volume, V. First, we algebraically rearrange our formula by multiplying both sides by N and dividing by C, isolating V. Next, we substitute our given values. The numerator becomes 50,000, which is the total number of viral particles needed. Dividing this by the concentration of 1 million gives us our final answer of 0.05 milliliters, or 50 microliters. This exact volume ensures maximum gene integration without toxicity. After successfully calculating the volume and infecting the cells, the next phase of the experiment begins. The treated embryos are implanted into surrogate mothers. However, biology is unpredictable. Not all manipulated embryos will survive the physical stress of the procedure to be born alive. Furthermore, even among the live offspring, not all of them will successfully carry the integrated transgene to become transgenic founders. Because of these biological hurdles, scientists use specific efficiency metrics to evaluate the success of the experiment and understand exactly where losses occur in the pipeline. To track the survival rate of our embryos, we use the birth efficiency formula. Conceptually, this simply measures the percentage of implanted embryos that successfully develop into live offspring. In this equation, E birth represents the birth efficiency percentage. The variable B stands for the number of live births, and E represents the initial number of embryos that were manipulated and implanted into the surrogate mother. We divide the live births by the initial embryos and multiply by 100 to get a percentage. If the physical stress of the implantation procedure increases, the number of live births, B, will decrease, which directly lowers the overall birth efficiency. Let us see this formula in action with a concrete example. Suppose a researcher uses the retroviral method on 200 embryos, which are then implanted into surrogate mothers. After the gestation period, only 40 live offspring are born. We want to find the birth efficiency. We take our formula and plug in 40 for B, the live births, and 200 for E, the initial embryos. Dividing 40 by 200 gives us a decimal of 0.20. Multiplying by 100 converts this to our final answer of 20%. This means 20% of the embryos survive to birth, which is actually a very typical survival rate, given the physical stress of the procedure. While birth efficiency tells us about survival, it does not tell us if the gene transfer actually worked. For that, we need the transgenic efficiency formula. This metric calculates the overall success rate of the entire procedure, from start to finish. Here, E total represents the overall transgenic efficiency percentage. The variable T stands for the number of transgenic founders, which are the offspring that actually carry our new gene. The variable E remains the starting number of manipulated embryos. By comparing the final number of successful transgenic animals directly to the initial number of embryos, we can see the true yield of our experiment. An increase in successful gene integrations will increase T, thereby raising the total efficiency. Let us complete our evaluation of the experiment. Out of the 40 live offspring born from our 200 initial embryos, genetic testing reveals that only 8 of them actually carry the integrated transgene. These 8 are our transgenic founders. To find the overall transgenic efficiency, we use our formula, substituting 8 for T and 200 for E. Dividing 8 by 200 gives us 0.04. Multiplying by 100 yields our final answer of 4%. This means that for every 100 embryos injected, the researcher gets 4 transgenic animals. While 4% might sound low, in the field of animal biotechnology, this is considered quite efficient and highlights the reliability of the retroviral approach. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that the retroviral method brilliantly hijacks the natural infection machinery of viruses, turning them into highly efficient delivery trucks that insert exactly one copy of our desired gene into the host DNA. Second, we saw how crucial the multiplicity of infection formula is for balancing successful gene transfer against cellular toxicity. Finally, we explored how birth and transgenic efficiency metrics allow scientists to track exactly how successful their procedures are from the initial embryo stage to the final transgenic founder. You now have a solid understanding of the retroviral method in animal biotechnology.